So I want to thank everybody here for coming to the Marine Institute Graduate Society Seminar Series. This is going to be recorded and uploaded to our YouTube channel where you can find previous seminars. So before we start, we want to provide a land acknowledgement. We respectfully acknowledge the land in which we gather as the ancestral homelands of the Beothuk and the island of Newfoundland as the unceded traditional territory of the Beothuk and the Mi'kmaq. We would also like to recognize the Inuit and Nunatsiavut and Nunatsiavut and the Innu and Nitassinan and their ancestors as the original peoples of Labrador. We strive for respectful relations with all peoples of this province as we search for collective healing and true reconciliation and honor this beautiful land together. So today is going to be a very exciting seminar. We are joined by Dr. Daniel Pauly. Dr. Pauly is a world-renowned fisheries scientist who is currently a Killam professor at the University of British Columbia's Institute for Oceans and Fisheries and the principal investigator for the Sea Around Us initiative. He has contributed to many concepts, methods, and software such as the Ecopath modeling approach and FishBase, among many others, which has been documented in over 1,000 scientific and general interest publications. He's also received numerous awards, including the Tyler Prize for Environmental Achievement, and the International Cosmos Prize, and many more. And for this seminar, Dr. Pauly will be presenting Fish Must Breathe, Introduction to the Gill-Oxygen Limitation Theory, GOLT. So with that, I will now pass it off to Dr. Pauly.
Hello, I cannot see you, and you will very soon not see me either. And I just want to warn you that I'm in West Africa right now, and the internet is not very stable, so I might disappear. I have not been kidnapped by aliens. You just wait 2 minutes and I get back on. So now is my presentation, and it is about fish that must breathe. And the Gill-Oxygen Limitation Theory is something that I did actually 50 years ago as part of my dissertation. At the time, it was only an attempt to understand what was happening with tropical fish and how they grew, because at the time it was very difficult to estimate how they grow. Daily wings were not available, nothing, and good method for length frequency analysis were not. So I wanted to understand how fish grow. And I worked on this for 50 years, and this has now solidified to a theory. Now, the first thing to realize is that when we ascend a mountain, we get less and less oxygen, and at 8,000 meters, there is simply not enough oxygen for us to breathe in the air, and we need to have oxygen tanks. Now, fish live at this oxygen, have oxygen in the water corresponding to about 9,000 meters. That is, if this were in air, we would choke to death. And in the water, there's even less than that. So the problem that oxygen is not available find itself in all parts of fish biology. But the first and most important is growth. And we will talk briefly about growth because you will then see what is misunderstood. Usually, in most textbooks, authors draw the grow fish in terms of length, and the inference is that fish grow rapidly at first, and then depending on how they reproduce, or they will then grow more slowly, and you have various options, and that is known as the reproductive load hypothesis, though it's not even perceived as a hypothesis. Here's another version of it, where the growth is in length is viewed as interrupted, so to say, by reproduction, and depending on the strength of that reproductive effort, the growth cease at certain length. But this is all in the books, in textbooks, that what I'm learned, and this appears to be something that you don't even realize is a hypothesis. Therefore, the ratio of, because of this, the ratio of length at first maturity, LM, to the maximum length, or the asymptotic length, the length that is calculated to be the last that the fish reach after a long, long time. This ratio is called the reproductive load, because the fish are supposed to, as they reproduce, to not be able to grow. So this forces a relationship between LM and Lmax. This is known as a Babaton-Holt invariant, which is not invariant in the first place. But it doesn't really matter, because both of them, this graph that I have shown you, are optical illusion. Because growth does not occur in length. It's a process involving mass. And if you express growth in terms of mass, it turns out that as strange as it is, the weight at first maturity, in other words, how heavy the fish are when they first mature, occurs before the growth curve, has its maximum growth rate. The point of the inflection of a growth curve is about 30%, sorry, of the maximum size they can reach, about 1/3. And that's WI. But WM is always before in fish that reach more than 10 centimeters. In other words, all commercial fish have spawn before their growth accelerate. So what it means is that it's not possible that the maturity and the gonad production cause the growth to slow down, as suggested when you look at length curve, because in weight it accelerates. That is not a choice that I made of one case. You can see that in all fish that you will be familiar with. If you plot the age at first maturity, sorry, the size at first maturity, and the inflection point of the growth curve, the age at first maturity, or the size at first maturity is always before that. This is astonishing because it completely is incompatible with the assumption that growth makes the fish grow slowly, but more slowly, but it's never questioned. Okay. Another thing that doesn't make much sense is to view what we learn again in all textbooks, that the environmental input, for example, warming temperature or changing light situation, causes the maturation. In other words, environmental input has an effect on the retina, the pineal gland, which affect the hypothalamus, which affects through a whole cascade of hormones, the pituitary, which affects the testes or ovaries, and leads to gamete maturation. So this is called an hormonal cascade, and the hormonal cascade is understood as self-starting. In other words, there is the fish perceive this, and then they spawn, or they mature and spawn. But this is incorrect or incomplete, sorry, because some fish will start spawning at 20 years. But what do they do at 15? They experience the environmental input, but they don't mature. And there's lots of fish that go through several spawning seasons, but they don't spawn. A cod, for example, will spawn at 4 years or 3 years. Well, what do they do at year 2? They're not ready. Well, this is just a restatement of the problem. So that is a problem that ought to be discussed. Now, here's another way of showing the problem. You have a growth curve. There's supposed to be stimuli are supposed to happen at a certain age, but before that, the environmental stimuli are not perceived. Why are they not perceived? They are there, and this you could generate hundreds of examples of this. This is the problem. Now, to resolve this problem, the two of them, we take a big breath and we look at respiring when in water. The problem is that water contains very little air, oxygen, but 30 times less than air. And it's more viscose and it requires more effort to get it past your gills. And diffusion, molecular diffusion, is 300,000 times slower in water than in air. This is not contested. This is the case. This is why over time fish have developed enormous gills. If you compare a fish, ancestors of fish from hundreds of millions of years ago, you have maybe a little part of the head that was devoted to respiration, and this was so small that they could not get big. Now, at present, your standard fish like a carp or a cod, the head is full of gill, and the biggest fish, they have a head like a jumbo jet that is also a big gills. And not obviously there is the exception. The arapaima in Amazon, it's a big fish, enormous fish, but it is air breathing. It's breathing air. If you prevent it from getting to the surface, it will drown. So big gills are needed to get big because why? Because I will explain. And this is not a fish. This is a French car. It was very progressive in its time, and it has this radiator in front. You know that the purpose of the radiator is to cool the engine so it doesn't overheat. Now, imagine that you want a bigger motor and it needs more cooling. So you need to increase the size of the radiator. Now, how does the radiator works in the first place? Water gets in, see the left side, water gets in and flows through the radiator, through lamellae, and the lamellae are exposed to air, which comes from the front of the car, and the air picks up the heat. And behind the radiator, the hot air is useless for cooling, and it's vented. Now, if you want to make a bigger radiator because you have a bigger engine, you can make it higher, you can make it wider, but you cannot make it deeper. Because if you put a radiator behind a radiator, it will not pick up any, it will not cool, because all the cooling that the air could do is done in the first layer. So gills are exactly the same way. If you look at the upper right, when water goes through lamellae, the oxygen gets picked up. It's quite efficient. About 80–90% of the oxygen is picked up. So there is no purpose in putting any gills behind the gills. What it means is that gills can grow only in 2 dimensions. They can grow only as a surface. That means, but the body that needs the oxygen grows in 3 dimensions: length, width, and depth. So a fish, for example, a cod that is 50 centimeters, I cannot see you, but I would, if I could see you, I would ask you, okay, a cod of 50 centimeter weighs, say, 1 kilo. How much heavier is a cod of 1 meter? Double that much. I have no time, so I will tell you the answer. It's 8 times heavier. It's 2 times longer, times 2 times wider, times 2 times deeper. So it is 8 times heavier. It has this is double in the third dimension. So basically, we have a surface. Gills are a surface, and volume. The body that needs the oxygen is a volume, and the bigger they get, the worse the situation gets. So here you can see a carp that grows, and you can see that the gill area also grows as they get heavier. And in the larvae, the growth of the gills is actually faster than the growth of the body. And at about 1 gram, there is a transition, and the growth then of the gills stabilize at a value lower than 1. That means, as opposed to things growing to the ratio of gills to body size being 2/3 to 1 or 2 to 3, it's 0.8 to 1. So basically, this parameter, 0.8 here in this case, is the rate at which the gill grow relative to the body growth. And you can see, you can estimate it using gill surface area measurements or using oxygen consumption, because oxygen consumption is quite proportional to gill area. And you can see that what gill area per body weight could be also respiration per body weight. And in the middle, the insert, you see that the gill area per body weight actually declines as the fish get bigger. And it declines to a level where the fish doesn't get enough oxygen. So that the growth has to stop. So this parameter D, this exponent of surface proportionality, this exponent is very important, and it is lower than 1. It has in the adults. And you can see here different estimates of D of this parameter. That is 0.8 in cod and in typical commercial fish. Actually, in smaller fish like guppies and little gobies, this is 2/3, like you would expect from a surface, real geometric surface. But actually, in most fish that are bigger, the gill fold in themselves. It's called invagination, and the result is gills that almost keep up with the weight. In tuna, D is equal to 0.9, and this results in gills, gill lamellae that are extremely close to each other. 160 gill lamellae per millimeter in tuna. It's extremely tight, but the tuna manage, by swimming fast against the water, to push water through this very thin layer between the lamellae. But most fish don't do that, and freshwater fish cannot do it because there is always grits. Is it grits? Silt in the water, so that it would plug the gills. So D is equal to 0.5, 0.8 or 7.5 and so on. So is always smaller than 1. So So if D is lower than 1, and the fish then stop growing at a certain weight, because the oxygen they get is just enough for growth, for living, for maintaining themselves, but not for growing anymore. So they can become bigger if they find a trick to reduce the maintenance metabolism. And so, for example, a fish that lives in hot and warmer water that goes deeper and reduces metabolism only by 10% here, it will be able to double its weight. Simply by reducing its metabolic rate, it's capable of increasing its weight by almost double. Now, here's another case where the opposite case, where you have a fish that reaches a certain size. If you increase the temperature, the maintenance metabolism will go up. We will talk about the reason later, but it is clear that when it's warm, fish and other water-breathing ectotherms need more oxygen just to maintain themselves. This forces them to be smaller. So that explains why in global warming, you will have colder, smaller animals, smaller fish, for example. Now, this one is what I just told you was well known as a phenomenon. The explanation was not necessarily there. Putter, for example, in 1920, produced a figure on the left up that showed that the predicted growth curve that you should obtain for a theoretical fish at 20 degrees and 0 degree. And this theoretician was exactly right. For example, if you compare the cod in Iceland, where the water is colder than in northern France, you can see exactly this type of growth curve. On the right side, you see European sardine, lots of them, and sardine from the North Sea and the Mediterranean. And in earlier, the North Sea is blue, the Mediterranean is red, and you can see the enormous difference. In earlier years, my colleagues, the colleagues who did this work, would have postulated that the growth of the North Sea one is bigger. They get bigger and they grow better because there is more food there. But actually, it's not the reason. The reason is that the temperature is different. Now it gets a little bit tricky. If a fish on the left, the A fish, spawns or reach a certain size, let's call it Wmax 1, then since you have seen it matures at a smaller size, let's call it Wm1. So this will correspond to metabolic level, oxygen consumption level, that are different. And Qm1 and Q maintenance 1 have a certain ratio to each other. The ratio can be computed in an empirical case. Now let's look at the left side, right side. On the right side, the fish stays smaller, and then it should, if it has any kind of sense, it should spawn also at a smaller size. And we know that it is what is happened. If a fish reaches 1 meter and spawns, say, at 30 centimeter or 50 centimeter, then if it reaches only 50 centimeter, it cannot spawn at 50 centimeter. It has to spawn earlier. So QM2 will be will be higher than QM1 and will be also higher than maintenance 2. Now, is the ratio between the two, QM2 and Q maintenance 2, the same ratio as in A? It turned out it is the same ratio. And it is the same ratio not only in one example, but in all teleost fish, all bony fish. All bony fish have the same ratio. And this is an astonishing thing. I wrote about it in 84, and with 34 species, that is A. And I kind of not forget about it, but I didn't write about it anymore. And then somebody else did 51 species, that some guys from Idaho State, I don't know them, that they did for three species of salmonid, freshwater salmonid, they got exactly the same slope. And then we did it for in sea for Turkish fish, 96 fish, we did it for Chinese fish, and we did it also for tilapia all over the place. And the papers are all written up, and this is an astonishing thing that basically all fish use the same threshold to determine that they're going to spawn, and this threshold is linked to the increasing difficulty in getting enough oxygen. So, well, some of you like the sturgeons, and they also work, and all fish. Here I've merged them, and then the crazy thing is that it also works for crab and crustaceans and so on, and so they are statistically all the same. And I forgot in this case for the ketognats to show the confidence interval, but the confidence interval are all overlapping. So basically all fish, all bony fish, all crabs, lobsters, and so on, plus a huge amount of other invertebrates have the same threshold. Now that gets very interesting. So basically, one can formulate the following hypothesis. When the maintenance metabolism, when the day-to-day metabolism rate gets close to the maintenance metabolism. In other words, when you as a fish more and more often get respiratory issues, you get out of breath, this is called hypercapnia, too much carbon dioxide in the blood, and it results in a low pH. A low pH of the body. The tissues of the body are have a lower pH, and this is a necessary condition for spawning. This is what triggers the environmental stimuli. In other words, when the message comes, I'm not gonna grow much anymore because I feel that I'm getting problem respiring, then it's time to look at potential mate, to look at what is this business about spawning season. And you look, as a fish, you look at the spawning season, and then it works. Then it is triggered and they reproduce. So that's why the fish reach maturity when they do. When when a certain size is reached. But that size is not an absolute size, it's a relative size to the, this is relative to the size that they can reach. And this size that they can reach is determined by the environment. If it's warm, they reach a smaller size, they have to spawn early. So with this, we have, I thought we have this thing sorted out, and I gave a master student of mine the job of tidying up also this story by doing the sharks and the rays. And so she assembled a huge database of sharks and rays and chimeras, rabbitfish, that the chondroichthys, whatever, elasmobranch, and she did it. And this was significantly lower than for fish and invertebrates. So because fish and invertebrates have the same value, I couldn't say, oh, that's because the sharks are different because they are sharks. That doesn't work, because the invertebrates are more different from the bony fish than the sharks. So one has to find the reason why the sharks are doing that. And basically, I mentioned that basically when your respiration level goes down, then you have inside your body a low pH. Incidentally, those of us that are not familiar with running, if we run, we have lactic acid in our body, right? In our legs, that's why it hurts. That's a low pH. And if you have too much of that low pH, you can die. This is the reason why mammals cannot run all the time. And that is also the reason why tuna that are played on a hook and line are not, if they are played too long, the meat cannot be used as sashimi because it gets destroyed by the low pH. Anyway, so if the low pH is the reason why the signal is sent to the brain to spawn, and you have a body full of urea, urea is a scientific name for piss. So if your body is full of piss, and this is a base neutralizes low pH. So basically, we hypothesized that basically because the sharks and marine sharks and rays have the body full of urea, the urea neutralizes the low pH, and they don't feel the need to reproduce so early. Therefore, they reproduce at a bigger fraction of the maximum size. That was known before, that they reproduce at a, but, well, okay. So this hypothesis is kind of wild, but it turns out that in Amazon, there are rays that do not use urea. And here we are, the marine ray, the same as the sharks, the urea-aligned rays in between, they are in between, but the freshwater rays, they are near the situation they are near the ratio for the bony fish and invertebrates. That means it is really urea which has this effect. And another hint of it is that the coelacanth also has this late maturity and is full also of urea. So with this, I will just do a little thing just for a second. I will just check that you that you hear me, because you would feel perfectly silly if you didn't. Are you okay with hearing me?
Yep, we can hear you, Daniel.
Okay, all right. So I will, if this story that oxygen then rules the growth and reproduction of fish, what else can we explain with it? Well, among other things, we can explain why the female, 80% of the species of fish have females that are bigger than the males. Now, the story, you remember the story at the beginning, that reproduction is supposed to have a negative effect on growth. Well, that means the female should be smaller, because the female should have a much bigger reproductive effort. This is how a female is actually defined, by the reproductive effort. And yet 80% of the species, you can check it out in FishBase, are bigger than the males. So what is the difference? The difference is that they are a little bit more quiet. And by being a bit more quiet, they can devote more of their oxygen to growth. And that's it. That's all there is to it. And another case, if you take the same species, the same condition, the same everything, except that one is domesticated and the other is a wild tap, the wild tap doesn't grow well, and the domesticated one grows very well. For example, in this graph, you can see the difference. It would be much bigger if it were expressed in weight between a domesticated char and the wild type char, and the difference manifests itself in the wild type being skittish. For example, when it sees somebody coming with food, they will split because they think it's a predator. The domesticated one will come and calmly eat. And the skittishness then cause the energy that is in the food to be burnt for running around, and the oxygen that is available for darting about, whereas the domesticated fishes are quiet. And another one is it's work that is done. To test actually the hypothesis, it's growing tilapia on the 3 temperature regime. Oxygen regime: normoxic, slightly hypoxic, very hypoxic, 90% and 80%. And basically, obviously, they grow better when they have more oxygen. But what is important is that the one that had no little oxygen spawned at smaller size than the one that had lots of oxygen. In other words, when you feel that you're not going to grow, you better reproduce. And so this experiment completely validates what I've told you just a minute ago. And also, when I formulated this theory, and I didn't know the fact here that fish that are young are full of oxidative enzyme. In other words, enzyme that require oxygen to be there in the body, whereas old fish, big fish that are old, have a lot of glycolytic enzyme, which means that the tissue operate often under oxygen, under missing oxygen. And the interesting thing is that the authors, you could not understand what the heck was happening, and you can see it in the title, the violation of the metabolic size scaling paradigm. Glycolytic increase in large fish. They described in the paper, they're surprised at this, and they attribute it, they say, probably to a surface volume problem, which what it is. interestingly, nobody else can explain that, but the GOLT, this theory here, can. Now, another thing is, all of you will have heard that there are daily wings in the otoliths of fish and invertebrates. But the daily wings are visible only in larvae and young specimen, not in old ones. And there is no explanation for it except the one that activity level that vary in the course of a day. There is an active period, inactive period, result in a difference in pH. When you are very active, you run out of oxygen, then your pH drops. If a low pH will obviously result in a different calcification or in different crystal deposition in the otolith than when you have lots of oxygen and high pH. So the alternance of activity level result in an alternance of pH level, which result in daily wings. And there is no alternative explanation, no alternative explanation has been provided. And not only does it explain why the larvae have it, but it also explains why the adults don't have such rings, because the adults are always in a low pH situation. There is no more difference between day and night. They have to be less active all the time because they all the time have low pH, and therefore there will be no rings, daily rings produced. And in fact, you cannot detect daily rings in older specimens. So another thing that you may know of is that the food conversion efficiency declines with body weight. It's linear when you plot it in logs, and that is a well-established fact. And in aquaculture, that's the reason why you don't keep your fish too long, because when they get too big, they eat and eat and eat, but they don't grow. And that is also the reason why in aquaculture, a huge amount of money is invested in getting air into the water, in getting oxygen into the water. And you can see here, these pictures are from China, the money that is invested and the energy that is invested in this oxygenation device, which must run all the time, and which makes the food that is provided be assimilated. It would not be assimilated before, it would not convert into growth. The funny thing is that the practical people, the aquaculturists, know that. The textbooks of fishery science don't. Now, this is going to be a little bit tricky to explain, but I will explain. I will try. So in A, you see the food conversion efficiency of fish declines essentially from 1 or under 1 in larvae. They have a huge conversion efficiency of the yolk sac into growth at 0.9 or something, and to 0, and at 0, at maximum weight, they don't grow anymore, but they eat. So the food conversion efficiency is 0. So A describes the decline with age, but actually is decline with size. And now the following happens when fish spawn. From 1 minute to the other, they lose weight. For example, if they spawn 20% of their weight, which cods, for example, do, they immediately become lighter, but the head doesn't change, right? And so the head provides them with oxygen as before. So that means if they have a smaller weight, but the same gills, it's like they are getting younger, and the food conversion efficiency increases. if they eat a lot during that period where the food efficiency is high, they will grow faster than would be predicted if they had not spawned. And what happens is that this mechanism is such that the fish at the end of the season, well, the growth season, has recovered the weight that it would have gotten if it had eaten while during the reproductive season, which they don't. During the reproductive season, while the eggs are maturing in their body, they don't eat. So they should lose weight, but they have lost weight. But then, when they get rid of these eggs, the food conversion efficiency increases. So if they eat, they will grow like crazy. And at the end of the year, what happens is that the growth is exactly what it would have been if they had not reproduced. And that's the reason why the von Bertalanffy curve doesn't have to show reproductive activity to be correct. The curve represents the growth that happens, but it also implicitly represent also the loss and the gain that happens during and right after the reproductive season. This is what's just published a few years ago and a few months ago, and Ian Fleming of your university has provided us with photos that I should show here. That was beautiful because it showed a kilt. I didn't know that name even. That was a salmon that had an Atlantic salmon that had just spawned, and it was very thin, like an eel. And 2 months later, it was huge. It was round again because it had not only eaten, but eaten and converted a huge amount of this food in growth. And so here, this paper, I think, resolved, or the reference is not given there, resolved the issues of why one can fit the von Bertalanffy curve, which has no reproduction in it, and still account for it in a sense. And I will be pleased to send the paper to whoever is interested. So taxonomists have always known that tropical fish that covered larger area are larger at the cold part of the distribution, and they also live longer. And we can see that it's a well-known thing that fish, for example, also in Newfoundland, the big ones, they are found in deeper water, and the young ones are called inshore in warmer water. And this is also shown for Hawaiian fish, that by going into deeper water, they get into lower temperatures, which reduce the metabolism. And I've shown before that reducing the metabolism a little bit, for example, by 10% for most fish, will allow them to almost double their weight. And another thing, migration are usually explained in terms of wintering, feeding migration, reproductive migration, and so on. For example, in West Africa, there is all from here in the south, Senegal, in the north, Morocco, there is all kind of explanation for the migration that different fish do in A, but in B, you have a simpler explanation, and scientifically, it's always better to have a simple explanation. The fish want to stay in the same temperature, and therefore the temperature changes with the rapidly with the latitude, and the fish migrate because they have to stay in the same temperature. That is a classical case. But in the textbook about fish migration that exists, it's by Harden Jones, a classic book. There's not even temperature is not even mentioned as an explanation. So I'm almost done. So basically, all of this put together indicates that that in temperate, cold temperate and areas, you have a mixture of fish. Some of them, most of them will be cold, cold. I have a cold water affinity on the left, and in the 70s you had this fish. In the 2000 you have fewer of them because they have gone further north or further south, depending on which hemisphere you are in. And in the future, we will have mainly warmer fish. In the tropics, for example, here in Ghana, where I am, and other places, you start the situation in the 70s, say, with tropical and subtropical fish. The subtropical fish leave, and you have tropical fish, and then in the future, all you have is a few fish left because it's getting too warm, and that is available as a paper. But basically, this mechanism is understood only if you realize the extreme sensibility of fish to temperature. Now, asymptotic growth is in fish is the rule, and it's basically due to the fact that growth rate is a function of two processes: one synthesis, one destruction of body substance. And the synthesis requires oxygen. So the factor D, the exponent D, makes this grow at a smaller rate than the W, than the KW, which is the destructive, the negative element, because it's also known as catabolism. Because this has no exponent, meaning the exponent is 1, and basically anything that grows according to weight will grow faster than something that grows according to weight to a power less than 1. And so whatever the starting conditions are, there will be a point at which the two terms become equal. At this point, the fish cannot grow anymore. Now, what is the negative force there, the destructive element? Well, I call it spontaneous denaturation. And spontaneous denaturation is actually due to globular protein like hemoglobin and other protein, spontaneously falling apart. They do that without any input from outside, because essentially Brownian motion, the fact that things vibrate at the molecular level, is enough to break them apart. So basically, say let's from 4 degrees, the temperature of the water, when the temperature increases, there is less oxygen in the water. But what is more important is that metabolism go up. And why does it go up? Because this denaturation. We will not talk about cold denaturation. Well, I will actually. This effect at low temperature is B, the line B, is the fact that when it gets really cold, the protein also the nature. And basically, you can see, if you look at the typical globular protein, in this case, hemoglobin, it consists of chains of amino acids, right? And the amino acid form a coil, and the coils themselves form coils, and the coils, the supercoils, form a shape, and this shape is called a quaternary structure. And this quaternary structure is held together not by chemical bonds, but by very fragile hydrogen link and electrical forces. And this structure can be easily broken apart. And in most protein, they last only a few days. They have a half-life of maybe 1 day, 2 days, 3 days, a week, but then they have to be renewed spontaneously. And when the temperature increases,
I don't think we can hear you, Daniel. Oh,
the body to gradually fall apart.
if you could just, sorry, we just lost you for a few seconds. If you could kind of just back up 30 seconds to where you were speaking before.
To there or there?
Yeah, so kind of like halfway through your conversation, during your discussion. Nope, you're good. Just go forward.
This?
Nope, one more.
Okay, so basically,
Yeah, right here.
what I was explaining is that all protein consists of amino acid chains, right? And the amino acid chains form a coil, and the coil form supercoils, and the supercoils form a structure, globular structure. And this globular structure works like a key in a lock. It works, the enzyme, for example, work because of this shape. But this shape can very easily be lost because it is kept together not by chemical bonds, but by H bridges and so-called van der Waal forces. It's very weak electrostatic means to keep these things in shape. So basically, Brownian motion, random motion in the water, and cells, living cells are water, right? Are watery. They can easily break apart, and you have a situation where all protein, all of them, have a half-life of about a few days, a few hours, some of them, but a few days, and they have to be renewed, this protein. They have to be resynthesized, because once they lose the quaternary structure, it's lost. They become random, so-called random coils. They have to be resynthesized. So basically, you have the situation where all protein in your body fall apart, and they have to be resynthesized, which costs oxygen, which uses and costs oxygen. Therefore, you have one thing that is volume related, and the other thing is surface related. And therefore, if the volume related problem increases, then you get smaller, because it's the only way you can survive. So the Gill-Oxygen theory, as presented here, is coherent, and it has survived numerous empirical tests. But it is not intuitive to mammals like us, where food is important and breathing is easy. In fact, athletes, free divers, and mountaineers are the only people concerned about getting enough oxygen. For fish and other water-breathing animals, getting enough oxygen is as big a problem than getting food. And what I've told you is largely not intuitive. And in fact, there are quite a few physiologists who disagree with me. Some of them do not understand the problem with surface volume, and some of them argue about me not understanding about maintenance, metabolism, and everything. But basically, this theory explains a lot of stuff, and we will have to modify some textbooks, but because the nonsense that I started with presentation with is really bad. And the nice thing about the GOLT, if it is correct, and asymptotic growth always occurred also in the past, then you can force the asymptotic growth onto fossils, and so I have done this on using an old method that I had developed to analyze tropical fish on trilobites that are 420 million years old in the Ordovician, and that was my first paper on paleontology. And I have another one that came out today about some animal in the Cambrian, also having a growth that is asymptotic. So basically, I would be pleased to talk with anybody who is interested in these ideas, and I will send papers. I will send papers. Also, my papers can all be had by going to the people of by clicking on people when in a bar of the Sea Around Us website. You click people, you can see all my publications are there, or you can write me at oceansubc.ca. That's it.
Perfect. Thank you so much, Daniel.
So we can have a discussion, or this is too long?
Yeah, do you have time for some questions? I wasn't sure if there was any more slides after your proteins and talking about the peptide chain, but that was the last slide that we saw. I wasn't sure if there was any more.
Oh, you didn't see the trilobites?
No, we didn't.
Oh, shame. So basically, I said that if this aggregate is the protein spontaneously fall apart, which they do, which biochemists are very clear about that, then you don't need any energy for the negative term of the basic equation, only for the positive synthesis part. And the synthesis requires oxygen. So you have oxygen limitation because the power weight by which it grows is less than the power of 1 that requires rebuilding or resynthesis of protein. So basically you have an imbalance, and the more temperature increase the number of protein that fall apart per unit time. So basically, we have an explanation at the biochemical level for the observation that fish get smaller at higher temperature. So it's not only a descriptive thing; it is a mechanism. A mechanism is provided, and with that mechanism, we can explain a lot of observation about fish and invertebrates and sharks. And at the end, I said that if this is correct, all of this, then basically growth always has to be asymptotic of adults. And if it is asymptotic, then you can force it you can force it onto fossil data, length frequency data for fossils, and then you know how they grew even 500 million years ago.
Mhm.
So,
Perfect. Thank you so much. Do you have time for questions by chance, or?
me, yes. Yeah,
Okay,
yeah.
perfect. So we have a bunch of people here in person and online, but I'll turn around the camera. So let's see everybody. Started that.
Okay.
So
I
yeah,
was really worried about the internet, and I'm glad that I survived it.
yeah, there's no issues whatsoever.
Good.
Is there any questions? John?
Yeah, really interesting talk. Great to see all this put together using examples from physiology, ontogeny, experimental studies. And my question was written before you talked about the trilobites. But I wondered, given that we know from fossil evidence about giant invertebrates and giant fish from the deep past, mega sharks and things like that, do you have a sense in terms of whether those oceans were sort of sufficient with oxygen to support them and be consistent with this theory?
I have a big problem with big sharks. My thing is I have a big problem with megalodon because it is too big for my liking. You see, whale sharks, the biggest fish is whale shark, right? And they live in warm water. Somebody said, you can't be right because they live in warm water. Well, it turns out that whale shark spend half of their time in deep water because they go yo-yo wise in deep water, hot water, deep water. So this is, I have a colleague who call them cooling dives. And lots of fish do that. They cool themselves by going into deeper water, come to the surface where the food is, and then again go down. So basically, if megalodon did something like that, they can even have done the opposite, which is to live in cold water and attack from below from time to time a big animal that live at the surface and then go back to cold water. And if they have done that, and we don't know if they did or not, they can have been bigger than the present day sharks. We have also, there is a teleost that got a bony fish, extinct, but it's called Ledistis problematicus, and it was as big as a whale shark, but it was quiet also. It was a filter feeder, just like whale shark, and one can very well imagine that they were fishing, going yo-yo up and down. And to make you understand that the yo-yo thing is not something that I invented just to save myself. It turns out that in the Red Sea and in the Persian Gulf, there is no adult whale shark. There is all kind of juveniles, but at about 7 meters, 7 to 8 meters, they leave because the Persian Gulf is only 30 meters deep, and you cannot do cooling dives in the Persian Gulf. And in the Red Sea, the water gets warmer if you get deeper because there's all kind of volcanic stuff. So there is both in the Gulf of Aden and in the Arabian Gulf, at the mouth of the Persian Gulf, there is you can get adult whale sharks, but not inside, not in the Persian Gulf, because the adult cannot do the deep dive. So I'm happy with explaining that. And you know, a few, I think 1 year ago or two, there was in Discovery Channel there was some stuff about megalodon leaving, that that really lived, and then it was at the end they said, "Oh, this is all not true. We just made it up." And the director of the CEO of Discovery Channel lost his job for doing that because they pretended that megalodon still existed, and it wasn't true. Yeah. Anyway, so big fish are a certain problem, but you can then show that their adaptation that they have that makes them tolerate hot warm water. I should mention another one. The mola, they get very big, and they live in the tropics. It turns out that 60% of their body consists of fat, some oily stuff that is inert, that doesn't react, that doesn't need to be maintained, and that doesn't denature, and so it doesn't need to be looked after, so to say, in terms of oxygen. So this is how they can get big in the tropics. Yeah.
Thanks.
Any more questions?
Thank you, Dr. Pauly. My question was about your ratio between the weight at maturity over the weight at maintenance for sharks, rates, and skates. Well, I mean, for all of them really, but you have it going through 0-0, which biologically makes sense, but it made the linear regressions look like they were almost forced. And I was wondering if
they are forced. They are forced to 0.
Okay.
They are, and in fact, think about it. If I didn't force it to 0, there would be another parameter, and this other parameter, the intercept, would have no meaning whatsoever. So it could not. I could not interpret it, and we would, yeah. And another reason is we can think of very, very small fish that are tiny. The smallest fish are 1 centimeter, less than 1 centimeter, and they spawn at, say, this is a fish that grows to 6, say to 8 millimeter, and it spawns at 6. Then you're close to 0. You might as well force it.
Yeah, it reminded me of stock assessment parameters where sometimes stock assessment or fisheries managers will move it to where it biologically doesn't make sense, where it crosses an axis like growth parameters and maturation, where it crosses an axis at negative 1.3 years old or something like that.
Yes, exactly. This is why we force it to 0, because actually, if you had a good representation at all sizes, it would be close to 0. If we had the tiny fish was represented, and in some cases I do have tiny fish. In fact, there's a paper that we just are finishing, where the fish are called reef fish, and the genus Aviota or Aviota, the adults are 1.3 centimeters, and so you are close to 0 intercept. But that's a very good point. That's a very good point. I have to write everywhere. We force it to 0, and therefore it's not the regression. It's not the fact that these things correlate with each other. The important thing is the slope, the value of the slope, and this is one way to estimate the average slope, the average ratio between this. I could also estimate it without regression, but then I couldn't show it.
Yeah. Awesome, thank you.
More questions? No.
One more question. At the start, you mentioned the life history of variants or non-invariant of length and maturity as a function of, or over length, asymptotic length. And you said that that wasn't really invariant, that it changed with fish size. I wondered, have you looked at your ratio between the weight of at inflection versus the weight of maturity? Is that invariant and you're just, and in the past you've used the wrong currency, or are there really no invariants?
So I think that the value of Lm over L infinity or Lm over L max is relatively narrow. It goes from 0.4 to 0.8 in fish. But it's not invariant. They have called it a Beverton-Holt invariant, but it's not invariant. In fact, the small fish mature at a larger proportion of the size than the small big fish. That I'm talking about bony fish, and the smallest fish are almost all semilparous. They mature and they die. I'm not talking about salmon, Pacific salmon. I'm talking about a very small fish. They can produce only one batch of egg and then they die. But when you raise the Lm and L max to the power of the ratio at which the speed at which they gill grow, it turns out to create something that is invariant. That is this 1.35 value, which appears to be invariant in trilobites, in fish, and in invertebrates, all kinds of invertebrates, except in sharks and rays. And I have shown you also in coelacanth, I have shown you the possible reason why it varies in sharks. The point is that if you have something that looks like a rule, but it has an exception, you cannot say, well, it's different, because the sharks are not more different than crabs or shrimp. So then from bony fish. So you have to find what is it that really separates the sharks from bony fish and shrimp and crabs. And it turns out that this is this urea thing, which is a base, and being a base chemically is offset. I think offset the role of low pH, and low pH, I think, is the mechanism that makes the fish spawn, or rather makes them be aware of the need to spawn.
Perfect, thank you. I have a question for you. So in the paper, like Pauly 2021, GOLT and the critics, you were talking about GOLT always assumes that there's enough food to grow, and that GOLT will have to be integrated into bioenergetic bottles or vice versa. I was wondering if there was an update on that, or if you can expand.
The first paper that I have done that begins to look at food is the one that shows that the fish, when they are maturing, don't eat, and therefore they don't grow as they would if they ate, and then they spawn and lose 20-30% of their weight. In the tropics, it's a bit less, but let's say we are in Newfoundland. So the 20-30% of their weight is lost, and then they become young again because they have this bigger gill per weight area, per gill area per weight. So in the 2-3 months that follow the spawning, they grow crazy, they eat up, and they grow like crazy. And then they get back to where they would have been if they had not they had not reproduced. And this, in the literature, is known as compensatory growth. But compensatory growth is not an explanation, is like a restatement of the problem, right? They don't grow, and then they have compensatory growth, and then well, yes. But what is it, and why can't they be compensatory growth? And this compensatory growth is because not only they eat, not eaten, but they grow faster on what they eat. And that is the first big step that I have done in integrating the growth. Another growth and the GOLT, integrating the GOLT with the food. Another one is that I have daily conversation almost with Carl Walters. You know Carl Walters? He's my neighbor. We live in the same house. And I see him every day. And one time, about half a year ago, he saw a presentation like this one. And he says, I hate to say it, but it was not as bad as I thought it was. Because for 20 years that we are colleagues, he never wanted to look at the GOLT. And he is retired and he has nothing to do. And he heard it, and then we spend a lot of time trying to integrate the models with growth based on food with the GOLT. And I must say it's very, very difficult. It's very difficult because obviously you can have as a description, but description, the DEB model, for example, they account for everything with hundreds of parameters. The point is that you can, with hundreds of parameters, you describe, you don't explain. And so we found the only thing that we ended up agreeing is that it's not that it is not only larvae to which the GOLT does not apply because they are not oxygen limited, but also very young fish. So a cod fingerling is not oxygen limited. So it is food limited, right? So basically the discussion that we had over the half a year is to define the limit of the GOLT. And it doesn't apply to larvae because they are not oxygen limited, and it doesn't apply to very young fish because they're also not oxygen limited. The rest we cannot agree on.
Right, perfect. Thank you so much. Yeah, that definitely helped. I'm excited to see more information on that when it comes out. I don't believe there's any more questions. I don't believe there's any more questions. Oh, sorry.
What I'm saying is, I will send you this last paper that was published in Environmental Biology of Fish, where it's called the postponing growth acceleration. It's postponing growth acceleration, and that's where your colleague Ian Fleming, you know him?
Of him.
Yeah, where he supplied this beautiful eel-like salmon that 2 months later had regained its roundness.
Yeah, I would love if you like sent that paper over to the email that we've been conversing through. That would be really great. Thank you.
So, all right.
I think that's it. I don't think there's any more questions, and we thank you for your time, Dr. Pauly. It's been really great to talk to you and see some of the work that you've done.
Well, it's beautiful not to have been interrupted too much.
Yes.
Yeah. Thank you very much.
Thank you so much.
Bye.
Stop recording.